and now introduce the seminar speaker. So it's a pleasure to have Professor Rich Carthew, who will give us this seminar today. Rich obtained his, there's a echo. Good. Okay. Rich obtained his bachelor degree from Queen's University in 1978 in biology. Sorry, I should not say dates.
Don't say dates.
Okay, I'll not. Then he went to University of Toronto to get a master's in botany.
Botany.
That's good. And then he joined MIT for his PhD under the supervision of Philip Sharp, who got a Nobel Prize later. Rich, his thesis was in characterization of human class II transcription factor. But one curious fact about his PhD thesis, or his PhD time, is that Rich was a mathematician at heart. He didn't know, but he saw like with his co-author, Lance Trodish? Louis Trodish.
Oh yeah, the biophysics stuff.
Well, he wrote a paper with the idea of just writing. We have actually two co-first authors, and you sort of like initiate a trend in biology that such a thing actually exists. And for us in mathematics, authorship is just alphabetical. Rich already had this in his heart. So he was already bridging math and biology in that community. Early days. Anyway, so he was pivotal to introduce this idea of co-first authors in biology, which is now actually widely used. Okay. After grad school, Rich went to become a Helen Hay Whitney Fellow at UC Berkeley, and then he joined the University of Pittsburgh as a faculty member. In 2001, he moved to as a full professor to Northwestern, and in 2006, he became the Owen L. Kuhn Professor of Molecular Biology. Rich has made pioneering contributions to our understanding of gene regulation, using fruit fly to investigate the mechanism of RNA interference and the quantitative principles of animal development. Rich's seminal discovery of RNA interference in Drosophila was instrumental in establishing the universality of this biological process and support the creation of many powerful genetic technologies used worldwide. You know Rich as the first director of NITMB. His leadership was important and fundamental to actually just bring together this fantastic group of people and to create this space. I also personally in debt to Rich because he sort of like changed my research life. In 2017 or 2016, he just called me out of the blue, and I've never met Rich, saying like, "Oh, I want you to be involved in biology." And as a pure mathematician, I was like, "What is going on?" But I went to his office and I said yes, and then here I am. Okay, I think there is a version of the multiverse that I said no, and I just continued doing math, and I got the Fields Medal. But I rather prefer to be here. All right, I'm really thankful to you. Anyways, it is my true pleasure to introduce Rich to his talk. But first, Tiffany, come. We have a gift for you, Rich.
Oh, a photo.
You gotta undo it.
Oh, that's so cool. The symbol.
This is all fabric from the build.
Oh, wow.
I put it together for
Oh,
you.
wow. So that symbol actually was also kind of a description of the collaborative effort that we had in putting this whole institute together, wasn't it? Because there were many iterations of the symbol. Let's try this, let's try that. And it was really sort of like a team effort when we all kind of came to the conclusion, this might be the best, or we were running out of time. We got to go with it. Although I think Bill Katz still prefers the mouse. Thank you very much.
You're welcome.
It's Carthew.
Thank you. So Tuka, my forays into math began with my first paper, my first published peer-reviewed paper, actually. So I was an undergraduate student at Queen's University, and I was working in limnology, which is the ecology of lakes, studying a cyanobacterium. And I got very interested in the physics of it. And so my advisor sort of said, do whatever you want to do. And I took it off and did some experiments and also some theory. And so I'm sole author on this very obscure, and it's actually a terrible paper, but it's a hybrid because there's both a theory and then there's experiments which actually disprove the theory. And I left it at that. It was like, my idea is wrong, and here it is. Which kind of brings me actually to this talk, which is an interesting sort of like lesson on how that happens. You can have a theory or models about how things go on, and they can be sort of generally accepted by a field, and then some experiment comes along and overturns the whole idea. And, of course, that's how science works. But this story here is one such story in my own personal life of studying science. I put the very famous painting here for a particular purpose, which I think you'll understand as we go on later into the talk, but it's very appropriate. Okay, so the general problem, of course, is how living organisms generate complexity and structure from very simple disordered precursors. So, for example, these beautiful butterfly adults all arose from simple eggs with radial symmetry, such as that. And through the process of development, of course, one goes from simple structural simplicity through development to forming structural complexity, where we have three major body axes that are polarized, so there's polarity to it. So there's symmetry breaking involved, and of course, then there's also pattern formation. There's many, many things going on to generate such complexity. And this black box development has been sort of like opened up a certain amount for the past 40 or 50 years. Used to be very much a black box. So when I was that ancient creaky undergraduate student in the 1970s, I took developmental biology, and basically this is what I learned. It was a black box. Now we know a great deal more about it. A lot of people over the years, actually millennia, have been thinking about, how does this complexity arise during development? And that included mathematicians like Alan Turing. So in the early 1950s, shortly before he passed away, Turing sort of turned his attention to problems in biology. And this particular problem really fascinated him because he wanted to come up with some sort of simple, elegant mechanism by which complexity could arise. And he came up with the idea, a concept which he called a morphogen. It's basically a chemical. It diffuses to form a concentration gradient, and then cells within the organism respond to the local concentrations that surround each and every individual cell. So this idea then was elaborated upon by primarily these three theoretical biologists in the 1960s and 70s to explain, for example, pattern formation and axis formation in simple embryos. The concept is the following. So they came up with the idea that cells across a particular range of concentrations would respond uniformly to that cue and, say, become a black cell. And there would be this threshold of concentration below which they would not respond and become black. Rather, there'd be a different range at which cells would now respond and become, for example, yellow cells. And then yet a different range of concentrations where cells would respond and become red cells. The original idea was actually the French flag. I adapted it a little bit because this is the flag of Belgium. But the idea is the same, which is like basically you have this simple chemical which provides information based on this concentration gradient, which is obviously dynamic because there's a sink of it. Never reaches equilibrium. So that idea, which hatched itself in the 1950s, served as an inspiration for developmental biologists thereafter. And it wasn't until the mid to late 1980s that the first morphogen was found. And it's a protein called bicoid. And on the left is basically the bicoid protein in a fruit fly embryo shortly after the embryo has been fertilized. This bicoid forms a concentration gradient, and indeed, it activates transcription of downstream genes with funny names like hunchback and caudal, and they follow this kind of thresholding rule so that this single transcription factor can form patterns, right? Like a French flag, except this forms the head, thorax, and abdomen of the future fruit fly. So that's one example of how theory can really inform us about how complexity develops in organisms. But there's another aspect of complexity, which is sort of the beautiful regularity of patterns that you see across nature. So for example, the pigmentation of such mammals and birds is highly regular. And then if you look even closer, what you see is that the skin itself has a very regular array of the fur hair follicles or the feather follicles that basically coat this. So there's many layers of sort of like regularity to the organization of structures such as follicles and many others. And probably one of the most striking examples is in the sensory organ systems of various animals. So the sensory organs have to be arrayed across the body in very regular patterns because basically they're mapping the environment into the brain. So there has to be a really, really regular array of input. That regular array you can see, of course, on the facial whiskers of your favorite cat. But it also exists inside the ear, the inner ear with the hair cells and the regularity of their placement in order to detect sound. The detection of light by the photoreceptor neurons in invertebrates. Then the array of sensory bristles that cover insects. And again, we could turn to Turing because he came up with, again, ideas around the same time as he was thinking about morphogens and sort of came up with the idea of a two-component reaction-diffusion model in which the two components were an activator and inhibitor. They interacted or reacted with each other with this kind of formal reaction pattern. And of course, they were able to diffuse through space. And basically, he was solving these models with differential equations. And this is just a simple form of it. You see the diffusion term on the right, the reaction terms on the left side. And you can form those reaction terms or even the diffusion terms in all sorts of different ways. And here's just a couple of simple examples to be consistent with that formal interaction relationship between U and V. And you can tune the parameters. And, of course, all of you are very familiar with this. But it creates really amazing patterns when you do that. So here are just three such examples. These are standing waves or type 6 Turing patterns. These are type 5 or moving waves. This one's an interesting sort of combination of both a standing wave and inside the standing wave are traveling waves. And these patterns, especially the type 6 patterns, really fascinated biologists because they were able to look at that and go, wow, those really resemble things that are really regular in nature. Such as, for example, the pigmentation patterns on seashells or the pigmentation patterns on tropical fish. And shown here on the left is a natural version and on the right is a simulated version of the same thing using a reaction diffusion model to create that. So this concept of this very simple mechanism driving regular patterns became very, very sort of catnip for many developmental biologists. We're always on the lookout. But the problem is, very much how do you gain the evidence that this exists? Okay, so I'm going to tell you a story about our study of the compound eye in Drosophila and our attempts to sort of understand this. The compound eye of any insect is really beautiful. I would say the fruit fly eye is a rather shabby version of it, even though you can see the regularity and the organization of hexagonal lattice with each one of these being a lens which covers a simple unit eye. Now, if you take away peel away that lens layer and look underneath at the array of photosensory neurons, you see again, beautiful regular hexagonal array where each of these clusters are unit eyes. You can see that their organization and regularity is incredible. There's a line of mirror symmetry separating two chiral forms of these, basically segregating them to either the dorsal or the back half or the belly half of each and every eye. And that boundary runs along the midline of the eye, and it is perfect. There are no mistakes. In fact, I won't say there are no mistakes. The error rate in this structure is usually 1 in 1,000. If we look at the single unit eye, it's actually called an ommatidium, and it's composed of 8 photoreceptors, given the names R1, 2, 3, 4, 5, 6, and 7. R8 is a neuron that lies below R7, which is why you don't see it. They're surrounded by pigment cells that optically insulate the unit, and of course, a lens over top of them. Very, very stereotypical organization. And this is repeated approximately 800 times across the one compound eye. Now, this beautiful regular structure basically arises from a much simpler precursor. And that simple precursor is called the eye imaginal disc. It's composed of roughly 20,000 cells, and the primitive eye begins in embryogenesis, and then it's buried inside the body of the organism, which is why, of course, insect larvae don't have eyes, because the actual adult compound eyes exist embedded inside as an internal organ connected to the brains, so one eye connected to each hemisphere of the brain. And these imaginal discs grow in size in proportion to the growth of the larva as it grows. But the eye also begins to pattern itself at this stage. And you can tell that by basically taking this off and examining it. When you do that, basically the eye itself is composed of 2 epithelial layers, a very, very thin epithelium called the peripodial membrane, and you can think of it like tissue paper, and if you tear off the tissue paper, then you see the eye proper, the second epithelium line underneath. And when people did this for the first time back in the mid-20th century, they noticed this interesting groove or furrow that existed, and it was very vertical, right? So it's a line basically along the dorsal to ventral axis. That equator or line of mirror symmetry I told you about is orthogonal to it, okay? And if you take animals at different ages, dissect out the eyes and look, you see a groove in every one of them, except the groove is at a different position. For younger animals, the groove is here, and as the animals progressively age, the groove is further and further towards the anterior margin. Came up with the idea, a moving furrow. And that was coined, the term was coined the morphogenetic furrow. And we've been studying this now intensively for, 40 to 50 years, and we know a lot about it. So, first of all, it is a traveling wave of pattern formation. That's pretty obvious because here is the morphogenetic furrow. You can't see the furrowing itself. And you can see that these particular cells that are highlighted with antibodies that recognize photoreceptor subtypes begin to light up, and they are lighting up in a very regular pattern behind the furrow. So the idea is that this is a moving wave of pattern formation. And you can see it in this fixed specimen over here. Here is the furrow, right here. And you see these clusters of cells aggregating with one another tightly, and these are the developing ommatidia that emerge sort of on the wake of the furrow. And in front of the furrow, the epithelium is unpatterned. Now, if you actually sort of measure the distances between these particular units. So over here is an idealized schematic, but these measurements are actually from real data normalized to this distance here. And this is basically the coefficient of variation for each of those measurements. So first of all, it tells you that it's very, very regular. There's not a lot of variability in this. And then basically the distances between each of those fits with the Pythagorean theorem. So it is forming a right triangle. So the pattern that is emerging in the wake of the furrow forms a rhombic lattice, not a hexagonal lattice. Okay. Now, the evolution of the rhombic into the hexagonal occurs over actually many hours, if not days. So as those ommatidia that basically were born hours before the furrow had passed, those are starting to now become positioned, and especially in pupal stage, positioned regular to each other into hexagonal lattice. But the point I'm trying to make is that the triangular lattice is formed at the wake of the furrow. So some magic is happening in there. And this is the pattern at the late pupa that you see. It's really beautiful structure. Now, in terms of like molecular mechanisms and how things, work inside the furrow, we know a lot about that now because we've been studying this, and by we, I mean this field of people have been studying it for, again, close to 40 years. First of all, we know that the cells are basically lineage restricted in front of the furrow. They are fated to become a retinal cell, but they can become any one of the 25 different types of retinal cells. But then as the furrow approaches them, they go into this transient proneural state in which they're basically competent to become a sensory neuron. But only a fraction of those do and become these R8 cells right here positioned behind the furrow. And all the rest basically relax into this sort of modified retinal state. They're now competent to become one of 24, not 25, because the R8 fate has been determined. And then what causes the furrow to move, what causes this transient establishment of different fates is all controlled by various signaling molecules like hedgehog, delta notch, a BMP molecule. There's various transcription factors, and one key transcription factor is called atonal because it endows the cells with that proneural quality. And when it's maintained, the cells become R8 cells. So the field came up with a fairly obvious concept that the positioning of future ommatidia, a future column of ommatidia, is basically programmed by basically its interface with a newly established column, where the elements of the newly established column send out inhibitory signals that diffuse and are able to inhibit their neighbors here, but these guys are too far away. And therefore, they become competent to become the next column of ommatidia. And obviously, because I talked about reaction-diffusion models earlier, that is a very, very favored idea about how this works. And there are even basically molecules we can attribute to the activator and the inhibitor. Two inhibitors, one being scabrous, and the other a ligand for the epidermal growth factor receptor. These are thought to send long-range signals that inhibit atonal expression inside cells, atonal being a local transcription factor that activates within the cell. And then you can resolve the sort of uniform field of atonal expression to this sort of salt and pepper, or I should say spotted pattern of intermediate groups. And then through a process of notch delta lateral inhibition, one cell out of each of those intermediate groups is selected to become the R8. And the R8 then, what it does is it basically induces its neighbors to become all of the other cell types. So they're basically the founder cells of each and every ommatidium. So their placement, the placement of the R8, is critical for the placement of the future ommatidium. So we, and by we I mean Vasile Alba. This is a selfie Vasile took during polar vortex. I don't know how many of you were here for the Chicago polar vortex, but he was crazy enough, because he's Ukrainian, to go outside and take a shot of himself in front of the lake. Anyways, Vasile adapted a reaction-diffusion model that was developed by Francis Corson for another problem in fly patterning, but for this problem itself here. And he invoked the activator-atonal inhibitor scabrous, sending local signals that are basically distance dependent. I'm not going to go into the details of the model, but it works very well, and it's highly robust. It gives, in this case, with the parameters that Vasile chose, gives a hexagonal pattern, but you can tune these parameters and coefficients quite easily and get any pattern you want. You can make stripes, you can make rhombic lattices. It's very straightforward. I teach a systems biology course, and I have the students play around with this model in order to make different patterns. It's a nice little exercise. But you can see that this model, which is very simple, it's driven by the wave, which is hedgehog activating atonal, known as a fact, then inducing atonal as a field that then interacts with the inhibitor and creates this pattern. All right, so there is a lovely theory, and it's been sort of like talked about for, I think, since 2011, with Dave Lubensky and Boris Schraman first came up with the idea and invoked their first model. But there's a couple of problems that have been sort of nagging us. One of them is that this sort of moving wavefront of gene expression is also a mechanical wave. So there's morphogenesis involved. That morphogenesis involves the constriction of the apical domain of the cells that are present in the furrow, and by doing so, it causes a mechanical buckling in the epithelium. And that's the presence of this. So the model I just showed you that Vasile had built was a vertex model. There was no accompanying morphogenesis. So why, and yet it works. So why have the morphogenesis in the first place? And then the other problem, and this has been a problem that sort of at the dawn of experimental research and the development of the eye has nagged all of us, which is that it was impossible to take out the eye or image the eye living as it was developing. So you could take it out ex vivo and the eye would die. And you couldn't image it by sort of deep imaging methods, because these are not transparent organisms, and the technologies to be able to do imaging were not high resolution enough to be able to see single cells. So all of the experiments are basically done by sparse sampling of fixed tissue, meaning you take out a sample, you fix it, and then you do whatever you want with it and look at it. And you have to do this with different, say, stages and so therefore what you're not seeing is the real dynamics that are going on. Instead, you're inferring the dynamics through this sparse sampling. And this problem, of course, has been bothering the field for a long time, bothered me. Every now and again, I would try to get live imaging to work, and it would fail. And so Kevin Gallagher, a graduate student, joined my lab, and he was encouraged particularly by Madhav, to take this on, to try and tackle this problem. Can you get live imaging to work? And it was fortuitous also that Henry Sun and Natalie Dye, different locations of the world, were independently working on this problem of growing imaginal discs. And they came up with some chemical formula. They've generously shared it with us. And that was the launching pads, because Kevin could get his toehold in it. They weren't robust methods, but Kevin then worked on it for close to a year and got a very robust methodology of imaging. It's very simple, although it looks very simple on the schematic. You basically surgically excise the eye. You put it in some media, you hold it in place, you image it, and then you can follow it over time. The imaging part was done by confocal microscopy, and the particular molecule we were imaging was GFP fluorescence. This GFP was tagged to or fused to a molecule called E-cadherin. Think of E-cadherin as kind of like a cellular glue. It sticks the cells of an epithelium together, and it's only localized near the apical surface of the epithelium, around the boundaries of every cell. It basically helps to glue these cells together to keep them coherent and not break apart. So a very important molecule, essential for life. And this was an endogenous knock-in of GFP into it. So that allowed us to follow the cells because we were able to see the outlines of the cells. And then do surface pull down and imaging. And the imaging we were able to get, so this is a movie, basically 10 hours of imaging time. The record that Kevin got was 20 hours of keeping it going and developing. But because it's a slow process, right? So embryogenesis in Drosophila takes 24 hours. And so this would have been equivalent to half of embryogenesis. Many movies you see about embryogenesis occur over a 1 or 2 hour time frame because events are happening so fast. So we needed basically to keep this going for a long period of time. But it worked really well. And then, of course, we had to analyze what we were seeing. And to be able to do this, Kevin developed segmentation software that was able to segment cell tracking, and then basically had to manually correct. Manual correction, if any of you have done image processing, is the worst part of the job. So what Kevin did was do 3 rounds of improvement of pixel classification by machine learning. And the final version you got was actually a convolutional neural network that he trained the model for, and it's over 99.9% accurate, which meant he didn't have to do much correction. But this was transformative because it could allow us to track every cell in our movie for all frames. So what did it tell us? Well, it basically said that a lot of the inferences from those static snapshot experiments were valid. So basically the order of development or differentiation of the various cells, the photoreceptor cells, the cone cells, occurred at the right time, at the right place as inferred. The morphogenetic furrow moved at a constant velocity. The velocity was the same as what had been inferred by static experiments. The births and deaths of cells, so pattern of cell divisions, which are restricted to anterior to the furrow and in a wave posterior to the furrow, that was recapitulated. And then the development of that line of mirror symmetry I told you about, which occurs a few columns behind the furrow, we could see that as well, because the ommatidia literally rotate in the epithelial field counterclockwise or clockwise. But there was one key assumption that the imaging did not agree with. And this key assumption was one of the sort of foundations of everything in terms of our experimental interpretations over the years, as well as modeling. And that is that the cells basically the cells in this eye do not move relative to one another. These were based on clonal experiments where people have made lineage, basically cells that were marked that were lineage related, right? So think of sister cells and sort of a small family of cells. And then you ask, okay, later on, if you form this early during development, how does that group behave later? Are they all together still, or have they dispersed because they've migrated and moved? And the answer was they all sort of stuck together. They would change shape, but they all stuck together. And based on that, we said, cells do not move relative to one another in this particular system. And that was critical for, of course, our interpretation of all the experiments that people had done, as well as the model. So Vasile's model is a static vertex model. Cells are not moving. And it would predict that basically, if you knew the pattern, say, of these this column of cells, that these cells you would be able to predict. If you could go back in time, you could predict the precise locations of where those cells were. In other words, there was a rhombic lattice prepattern ahead of the furrow, and it was just waiting for that to get to grow into it. But that's not what we saw. Instead, when Kevin did that go back in time, so he identified the ommatidia in several columns at the end of his movie, and then he asked, where are those particular cells at the beginning of the movie? And this is where they are. And if you look at that pattern, it is not a rhombic lattice, it is a simple rectangular lattice in which not 2 columns are present, but 4 columns, 4 future columns of ommatidia are all compacted within that simple rectangular lattice. So how can that be? I'll just let you watch. So clearly you can see that these ommatidial cells, even though they're not ommatidia yet, they're moving with the furrow. The furrow is basically marked with this light gray, and you can see they move for a while, and then they stop moving with the furrow, and they bud off on the right side. So they move with the furrow at the same velocity as the furrow. So here is basically tracking each of those columns, all those cells in each column, and their position relative along the AP axis. So over time, they're basically moving anteriorwards, all with about the same velocity, but at different times they stop moving. And that is spaced apart every 2 hours, which is the time frame over which new ommatidia emerge from the furrow. Okay, so there's flow of cells. It's very regulated flow, and it's periodic. So in this exercise, what Kevin did was he's showing us the averaged velocity of cells, and the vertices of each of these triangles represents a future R8 cell or an already established R8 cell. So it gives you an idea of what the organization of the R8s or the ommatidia centers are. Then you can see how the triangles evolve in shape over time and how that correlates with flow. And I think you can see quite easily that there are periods in which there's clusters of high flow beside clusters of low flow. And that periodicity is very stable. So you can do a Fourier transform of all the data, all frames of the movie, multiple movies. That's what this shows you here. And for the cells in this bin, which is basically at this part of the morphogenetic furrow, very strong signal with the period length of around 14 micrometers. That is very close in distance between 2 different ommatidia in the same column. So the periodicity of the flow matches the periodicity of the ommatidia in terms of spacing. There's also periodicity over time. So the flow rates oscillate from high flow to low flow, then back to high flow. They oscillate with a period length of 4 hours. And then there's different basically phases of this oscillation pattern, depending on whether the ommatidia are on an odd even number column or versus an odd number column. So the odd number columns are 180 out of phase with the even. But all of them have the period length of 4 hours. So there's periodicity both in space and in time. So that obviously creates sort of like a bimodal behavior that you can very easily see with these 2-dimensional plots of R8 cells. So in the red are the R8 cells, And in the position of the furrow in that yellow zone, you can see there's basically a high flow population and a low flow, basically stationary population of R8 cells. Ahead of that, they're moving with the furrow. You'll also notice that in this zone and in this zone, relative to the furrow, the cells are actually moving towards the posterior. There's flow as well in those cells, but it's in the opposite direction. It's not towards the anterior side of the eye, it's towards the posterior side of the eye. And then it's sort of separated by places where there's little or no flow. And you can see that in this sort of one-dimensional plotting of, again, this is data from all of the time frames of our movies for all of our movies. And it's showing you basically the moving line average, and the 95% confidence interval is on here. It's so tight. This is so stable and regular a behavior that you can basically predict precisely where these transition points are relative to the furrow. So basically, you can see that there's a zone in which there's posterior flow, right? These numbers are negative, but then a zone surrounding the morphogenetic furrow where it's positive, and these two zones where there's no flow whatsoever. So of course, that implies there's a source and a sink of the flow. So the flow of cells is moving away from here, and it's moving towards here. A source and sink of flow. That would explain these flows. So what generates the flow? We spent a lot of time studying all sorts of different mechanisms, and our best candidate, although we still don't have definitive evidence that this is the way it works, is the following, that is based on pressure gradients that are created when the cells undergo contraction ahead of the furrow, right? So remember, the cells are sort of big and wide, and then they shrink in area, and then they grow back again, right, to create that buckling. So in the process of shrinking, there's cell contraction, and in the process of growing back again, there's cell expansion. And the zones of maximum expansion and maximum contraction coincide with the positions where the source and the sink of the flow occurs. That would imply that if the cells are expanding in volume behind the furrow, that then pushes their neighbors away from them, forming a pressure gradient. And that pressure gradient is especially strong because over here, there is basically a low pressure zone where the cells are maximally contracting. Okay, so that is our best explanation for what creates the flow. Might not be the only mechanism, but it is one mechanism. And I'll provide some genetic evidence later on that helps support that. But obviously, what generates the periodicity of the flow? Our first idea was that there was periodicity to the pressure gradient itself, so that basically there were zones of higher pressure besides zones of lower pressure, so you would get this, this periodic behavior. But when that would predict then that the posterior flow on the other side of the source would also have periodicity, but we did not see any evidence for that. Rather, it seems to be uniform posterior, and yet it's periodic anterior to the source of flow. So that made us think, well, perhaps there's obstructions or obstacles that are basically focusing and deflecting the flow around them, creating zones of high and low pressure. And then the placement of these anchors must be dynamic because it's alternating every 2 hours, low flow, high flow. What could those be? Those immobile objects. Well, turns out that when we looked at the mobility of the R8 cells themselves, they behave very different from the average in that they radically or quickly dropped their velocity to 0. They became immobile on this side of the source of flow. And the cells that were attached to them, the R25 and 34, they basically stopped decelerating, remained at constant anterior velocity. But this became immobile. So the idea is that as the R8 cells are basically formed, that they pin themselves in some way, which we don't understand how. They pin themselves or anchor themselves and become immobile objects, focusing the flow around them. And then you see at the source, they let go. So they let go of the anchor, and they basically flow in the posterior direction at the sort of ambient rate at which all the other cells are. So there's this transitory sort of immobilization which deflects the flow and creates the periodicity. And every R8 cell, as it's born every 2 hours, will form alternating anchor positions. Okay? So just to summarize the observations, hopefully it's made some sense. We think there's a source and a sink of flow. This causes flows of cells in different directions towards the sink, away from the source, that ommatidial cells, particular R8s, become immobile. That creates basically deflections in the flow, periodicity of high flow and low flow, and this is responsible for the evolution of the emergence of the rhombic lattice. So then, of course, that says, okay, well, you had this lovely model, very beautiful, very elegant, very simple. It works. There's no flow, obviously, here. Doesn't discount that there isn't reaction diffusion going on, but it obviously doesn't account for the flows. And then it made us think, what about these key elements? The inhibitor, for example. Is it really an inhibitor of scabrous, of a tunnel? So again, the interpretation of scabrous acting as an inhibitor is all genetic, based on static experiments. And this is sort of like the pattern of ommatidia you see in a scabrous mutant eye. So you can see that they almost form stripes of cells rather than a rhombic lattice of cells. It was interpreted that this was because the cells basically nearby emerging ommatidia were now sort of no longer inhibited. There wasn't that zone of inhibition. And so they were competent to also become ommatidia. And so you see these stripes. Classic sort of like neurogenic phenotype here. The other line of evidence was that we knew that scabrous is expressed exclusively in the R8 cells for 3 columns as they emerge off of the out of the furrow, and then it's shut off. Okay, so it's produced in the R8 cells, but it's not needed for R8 development. It's needed for this inhibition, right? And then structurally, it has the structure and also biological properties of being a secreted protein. So the idea is it's a secreted inhibitor. But is that the case? So Kevin, what he did was he took a scabrous mutant and he made movies out of them. So highlighted here are basically the R8 2534 that are emerging. You can see they form these very large irregular groupings. Some of the groupings will split apart, and this was well known. So the pattern comes off as stripes, but they then much later on will break apart into sort of pseudo-ommatidia. But you can basically see the same phenotype. But when we did the flow analysis of this, we saw a couple of interesting things. And one is that there's no periodicity in the cell flow in the furrow, in the scabrous mutant. Instead, the R8 cells basically no longer bimodal, but they're basically almost all stationary. And they're almost all stationary because the flow rate is greatly reduced in these cells. And in fact, all of the cells. Now, the loss of the periodicity, we first thought, okay, that's due to maybe R8 being unpinned. Scabrous is important for pinning R8. But that was not the case. It still seems to be immobile. But as I said, the rate of flow is reduced in the scabrous mutant. So shown here is basically the profile of the wild type versus scabrous. You can see that source and sink are actually closer together relative to the furrow than in wild type. But you can also see that the maximum velocity of flow is reduced by somewhat, and on this side as well. So we interpret this as basically, because the slope of this is less, basically the sort of acceleration of cells away from the source of furrow is reduced in the scabrous mutant. The acceleration towards the sink, though, as you can see, is pretty normal. And when we look to see if there was sort of evidence of a loss of the pressure gradient, what we observed was that indeed the maximum rate of expansion of cells at the source in a scabrous mutant were reduced relative to wild type, whereas the maximum rate of contraction in the sink was not. So it leads us to the following model, which is scabrous is actually regulating the expansion of cells. It's a signal from the R8 cells to regulate and trigger expansion of the cells. And in the absence of that signal, the cells do not expand as much. Therefore, the pressure gradient is weaker. There's no flow, there's no periodicity of the flow because of the weak flow in itself. And that leads to a scabrous neurogenic-like phenotype. Okay, so eye patterning involves a mechanical component. We think atonal is driving expression of scabrous in the R8 cells that helps to accomplish that. Now, does that completely eliminate the idea there's a reaction diffusion component to this? No. There is certainly the idea that this EGF receptor ligand could be acting as a reaction diffusion interactor with atonal to create the prepattern, the rectangular lattice, simple rectangular lattice prepattern. But it is this critical interplay between the chemical side of things and the mechanical side of things that generates the emerging triangular lattice. So basically, this system works like Kelly Slater, the world champion surfer. So that degree of control allows cells to flow with the moving wave, and then at precise times exit the wave in a very beautiful and regular manner. And that's in contrast to a more dysregular mechanism, such as an scabrous mutant, in which you don't get that highly regular control flow of ommatidia. And instead, you end up with that. I'll just end by saying this may not be unique to the Drosophila eye, this interplay between a mechanical wave and chemical patterning systems. So for example, in the developing epidermis of birds, so of mammals, you see a wave flow from either side of the backbone in the embryo. This moving wave also corresponds to a mechanical process that people are studying now involving cell density. There are mechanical waves, that is, furrows that are present in the developing brain, in the developing inner ear, and also during neurogenesis of vertebrate embryos, which is basically the very earliest stages of separating the nervous system tissue from everything else. So the fact that there are these furrows that coincide with fronts of gene expression patterning might suggest that this might be a more universal mechanism in which one can elaborate, say, simple rectangular patterns of chemical sort of interactions to create rectangular patterns to create more sort of triangular lattice patterns. And I'll end with that. The work was done by a single graduate student, Kevin Gallagher. Vasile Alba, I showed you his model. Kevin and Vasile were co-mentored by Madhav. We're indebted to Boris Schraman, who hosted Kevin back in the very early days and was a great encouragement for him to continue on with it, and Natalie and Henry for generously sharing their media. Thank you.
Questions? Beautiful movies and a very gorgeous talk, but I want to look inside the cells. You're kind of consider yourselves as bricks that do things, but I want to see what drives things there. For example, you have expansion of the cells. What kind of mechanical forces can generate expansion? I understand very clearly what can induce contraction, as you do, but how do cells expand?
Right, so I was showing you area measurements, and of course that could mean there's no volume difference. So the implication is to create pressure, you need a change in volume. The cells would actually have to expand in 3 dimensions, not just in 2. So there's a group in Dresden that has done careful, now after this work, after we published this work, has done careful measurements of this process in movies like us. And they do note that there is expansion of the cell volume at the source, and that there's contraction or shrinkage of cell volume where we said the sink occurs. So there is actually volumetric changes. That's one thing. Now, how do those volumetric changes occur? Of course, yes. So are there pumps that are regulating the flow of water, for example, into them?
So essentially, what you're thinking, that's a water flow inside the cell.
I think so, but we don't know for sure. There have been no mutations in such pumps that have been discovered from screens. One might have predicted, well, you might have been able to detect genes by mutagenesis, which would give scabrous-like phenotypes, which have not been uncovered. Now, that could be that these molecules are essential for life in many places, if they're general water pumps, and therefore we wouldn't be able to see them, in the eye, because the animals would have been dead long before.
You can drive her in the eye, and
yes,
and effect you get there.
Absolutely. So it leaves open the idea that scabrous is signaling, and our idea is maybe some sort of pumping system that brings water in, and then a pumping system that extrudes water from the cells. But we don't know for sure.
How about ectomizing and driving the contraction?
So that is the one thing we've looked at. So we've used squash GFP to be able to read out myosin activity. We see absolutely no correlation between actomyosin activity there in the source and the sink that would explain it. Also, no regulated myosin activity that would also explain the flow. So convergent extension, that was one of the obvious things we thought about. But there's no pattern to the myosin that is sort of consistent with a convergent extension process. And if we also look at the pattern of cell rearrangements over time, it doesn't follow with convergent extension, like T1 exchanges that are oriented in that axis.
I have a question about, so you gave this really beautiful account, which I really appreciated. And I'm a neuroscientist interested in learning and to what extent sort of a blank nervous system is structured through experience, and what extent sort of a structured nervous system accommodates the world. And I'm really curious about these two extrinsic forces, the mechanical and the chemical, patterning a sheet of cells, and whether any sort of structure in the cells themselves is required, or whether they're just sort of a uniform sheet that then is shaped by these external signals. Like, is diversity necessary at all in these systems through sort of either random processes or determined ones?
So, well, getting to your point about experience, one of the early ideas was actually that the development of this system was guided by experience, by light coming in to the primitive epithelium inside the body and helping to sculpt or shape the patterning of that. was discounted as an idea. Instead, it's more of sort of a program, a developmental program that is independent of experience. And it's only later in life where that really counts. In terms of sort of like heterogeneity in the cell states themselves, that's something that now people have done single cell RNA sequencing of in this system. And to be honest, all that data is consistent with the idea of what we put up here. Of course, there's heterogeneity, between the cells in terms of their transcriptomic profiles, but we don't see a broad, sort of collection of states on UMaps that then sort of focus into a flow. But it's a very, very difficult question to ask, especially when you're invoking sort of stochasticity into it as well. So, I mean, I don't know. I can't answer that question very well. My own feeling is that heterogeneity is important.
If you mutate atonal, or I guess you can't actually mutate the repressor, but if you mutate atonal, what patterns would emerge, or will the system still function?
So atonal, basically the name of atonal describes the condition. There's no sensory organs that are formed. Well, it's no hearing, right? So it's important for the hearing system. And then it was later shown to be very important for the eye system. So the absence of atonal causes no cells to become photoreceptors, right? Because you need that activator to put cells into that transient proneural state. And then once the cells resolve to, say, a cluster or a single cell, atonal actually shuts itself off, and other transcription factors take over the job. But because it's sort of like the pioneer, it's essential. So it's necessary. The inhibitor, basically the scabrous phenotype I showed you, and the ligand of the EGF receptor has a very similar phenotype. And in fact, a double mutant between the ligand mutant and the scabrous are additive, meaning they seem to operate independent of one another on atonal.
Okay, this question might be coming from like a lack of understanding, so please, yeah. So the fact that there's scabrous that's sort of driving the sort of shrinking and expansion of the cells, which is sort of driving the flow, does that eliminate the need to consider R8 being rooted as a pillar influencing that kind of flow? Like, does that remove the need to think about that? I don't know if my question makes sense.
So the fact that scabrous, R8 still form
Yeah.
in the absence of scabrous.
No, more like, I'm probably not putting it very well, so just.
So the R8s are still acting as pillars. It's creating many pillars, right? So because it's a self-fulfilling prophecy, the expansion of the cells is weak, and therefore the flow is slow, and therefore multiple cells can now adopt R8 fates. And now you have multiple pillars that impede flow even more.
Okay, gotcha. Sorry, I missed the part where the R8s are still acting as pillars.
They are still acting as pillars, yep.
Okay, any other questions? No, let's thank Rich again.